Okay, let's start. First of all, apologize if I'm not there in person, but I might be a close contact with a positive case, so I had to take the PCR test, and I'm still waiting for the results. So I really apologize, in particular for Jodi. It's so exciting to have an in-person speaker and and I'm here at home, so it's kind of annoying. But anyway, so let's hope for a negative result. It is, this is a seminar organized by my unit, and it's a pleasure to have Jodi here. So Jodi is originally from New Zealand, where she got a degree, an undergrad in neuroscience from the University of Otago. And then she decided to join the laboratory of the great Phil Munday at the IRC Center for Coronary Study in James Cook University in Australia, where she took a kind of a bold decision. So if you know Phil, normally if you go in his lab, you study fish, but it's similar to what we do in OIST. So the effect of fish on climate change. Jodi took this bold decision to focus on a cephalopod, which actually turned out to be perfect, because at that time there were not many people study the effect of climate stressor on cephalopod. Most people like us, we focus on fish for several reasons. But she was one of the first to show what she will show in a second about what we should expect on a cephalopod if we push in the climate towards ocean acidification or warming, in particular acidification. So also you should notice for the past year, Jodi was an official OIST visiting student, but she never visited until now because of course coronavirus, unfortunately. So she's supposed to spend a lot of time in OIST with us, but she just arrived yesterday. Well, Jodi, better now than never. So without further ado, I will give the floor to Jodi. For the question, for those people in the room, just please ask the question after Jodi is done with the talk. For those people online, you have two options, or you can speak up, or you can type the question in the chat, and either myself, Yoko, or Jodi will read the question, and we go from there, okay? All right, Jodi, thank you.
Perfect. Thank you so much, Tim. So as Tim says, I've been doing my research in Townsville in Australia and being a visiting research student here at OIST with the Marine Climate Change Unit. So it's really cool to be able to be here in person and present my research in person. So thank you so much for having me. And I'd also like to begin by paying my respects to the Bindal and Walgarukaba people, who are the traditional owners of the land as well as the waters where my research has been done in Townsville. So I'm going to talk about some research that I've done as my PhD, and my PhD has really focused on how climate change is affecting the nervous system of these tiny little squid here. So the nervous system is a complex part of an animal, and it's made up of these peripheral sense organs which connect to the central nervous system. And the main role of an animal's nervous system is to coordinate its responses to the environment. So for example, in squid, they use their eyes to detect movement of prey. And the information gathered by the eyes is sent to the central nervous system, where higher order processing is carried out to interpret that information. And then outputs are created and sent to effectors. So this, in this example, could be sent to muscles, which causes the squid to then strike at their prey. We also see the nervous system coordinating the release of digestive enzymes, which prepares the squid to digest its meal. So overall, we're seeing that the nervous system is really important in mediating an animal's response, both behaviorally, but also physiologically, to changes in the environment. And these changes in the environment can be natural. So this example of a prey animal swimming past the squid, but it can also be induced by human activity. So I'm sure you're all well aware that human activity is drastically altering the environments in which animals live, from climate change to habitat destruction and species overexploitation. And so an animal's nervous system is important to coordinate responses to not only natural changes in the environment, but also to these human-induced environmental changes. And for marine animals, a particularly important aspect of human-induced environmental change is ocean acidification. So human activity is resulting in unprecedented amounts of carbon dioxide, or CO2, being released into the atmosphere, and levels are projected to increase going into the future. The atmospheric CO2 is then absorbed by the ocean, And so we're going to see higher levels of carbon dioxide and a decreased pH in our future oceans, which marine organisms will need to deal with. So this process of rising carbon dioxide levels in the sea and the associated changes in ocean chemistry are known as ocean acidification. So a lot of research looking into the effects of ocean acidification has found that high levels of carbon dioxide can affect these wide variety of processes of marine invertebrates, including physiology, behavior, and life history traits. But this research has found a lot of variability in these responses to ocean acidification, with some taxa being more sensitive than others. And so marine invertebrates are really important for marine ecosystems. So they are vital for the ecosystem processes and are really also important for sustaining human societies across the globe, which means any effects of ocean acidification on marine invertebrates has this potential for far-reaching consequences on social consequences, ecological and economic. And as I said before, a lot of research has really focused in on these responses of marine invertebrates to high carbon dioxide, the physiology, behavior, and life history traits. But much less work has looked into the mechanisms that underlie these responses. And understanding the mechanisms of how high CO2 causes these responses is really important to help us develop these cause-effect relationships, to better understand this variability in the responses to ocean acidification, but it will also help us improve predictions of how marine invertebrates and ultimately the ecosystems they make up will respond as climate change progresses. And so looking at the nervous system is really important to better understand these mechanisms. As I said before, the nervous system.
Vincent said they will come at Spotify. They're going to be.
Okay, but just checking, still hear me, cool. So we have the mechanisms are really neurobiological mechanisms because the nervous system is really important in mediating those responses to environmental change, including ocean acidification. And so research that has looked at the neurobiological mechanisms is really understudied, but the majority of work has really focused in on fish, and it's concentrated mostly on this GABA hypothesis, which suggests that altered function of a specific receptor in the nervous system called the GABA-A receptor underlies the behavioral changes at elevated CO2. And because this research has focused on this GABA hypothesis, it's really left other mechanisms very little explored. And then in marine invertebrates, we have much less research. So the few studies that have been done suggest altered GABA-A receptor functioning might underlie behavioral changes in a conch shell in a clam, but not in a crab. But overall, we really don't know if the mechanisms identified in fish so far may be the same or different within marine invertebrates. And marine invertebrates themselves are highly diverse. So within mollusks, which squid, cephalopods belong to, there are over 110,000 different species, which means that the mechanisms could also vary between marine invertebrate taxa as well as between marine invertebrates and fish. So the overall aim of my research was really to look at the impacts of ocean acidification on the nervous system of a marine invertebrate and use this to explore the mechanisms within the nervous system that underlie those responses to high CO2. And to do this work, I used a cephalopod mollusk because they have a complex nervous system and behaviours that rival those of fish, which make them really a good model to look at the neurobiological mechanistic underpinnings. My study species was this little guy, the two-toned pygmy squid, Idiocephalus pygmaeus. And so these squid are found on the coastal waters year-round, around Townsville, where I did my research. And as you can see, they're very small and they have a very short lifespan of up to 90 days, all of which makes them really useful for experimental work. Previous work by Blake Spady in my lab has also found an effect of ocean acidification on a variety of their behaviours as well as reproduction, which created a really good basis from which I could then look at the mechanisms underlying these changes. So for my work, I focused on the central nervous system in the eyes. You can see in this microscope picture, we have the two eyes in black with the central nervous system sitting between and behind the eyes. And I did this so I could look at peripheral sensation, choosing the eyes because squid are highly visual animals with many visually mediated behaviors. And then the central nervous system also allowed me to look at that high order processing. Okay, so to start my research, I looked at the GABA hypothesis, as I said, is that major hypothesis in fish, to see if this is also happening in a cephalopod mollusk. So before I go into this, I just want to step back a little bit and go into the theory behind the GABA hypothesis. So the GABA hypothesis result is in relation to neurotransmission. So we have neurons or brain cells, and they communicate via electrical signals that pass between neurons by a process called neurotransmission. So when we have an electrical signal arriving at the first neuron, it results in the release of neurotransmitters or chemical messengers, which act on receptors on the second neuron. Depending on what neurotransmitters are used and what receptors are bound, this electrical signal can either be passed on or not. And so neurotransmission is really vital for all of the stages of the production of behavior. So it relays electrical signals from those sense organs up to the central nervous system, then around the central nervous system as that information is processed, and then neurotransmission also relays them back out to the effectors to create the behavioral response, which means that any effects of ocean acidification on neurotransmission has the potential to have very widespread behavioral effects. And so the GABA hypothesis relates to a type of neurotransmission mediated by the GABA-A receptor, which is a ligand-gated ion channel permeable to bicarbonate and chloride ions. So what we usually see in current day conditions is the neurotransmitter GABA binds to the GABA-A receptor, opening the channel, and there's a net influx of negative charge. And that usually has an inhibitory effect. It stops the electrical signal being passed along. So essentially, the GABA-A receptor is like a red stoplight. The GABA hypothesis was proposed by Nilsson and colleagues in 2012 in 2 coral reef fish species. And under high CO2 conditions, we see a drop in pH. And so water-breathing animals like fish and marine invertebrates will defend against this pH change by using pH regulatory mechanisms, which involves changes in the concentrations of bicarbonate and chloride ions, those same ions that the GABA-A receptor is permeable to. So this hypothesis suggests now at high CO2 conditions, due to the changes in bicarbonate and chloride ion concentrations, we now have a net efflux of negative charge, which has an excitatory effect. So the GABA hypothesis essentially states that there's a switch in function of the GABA-A receptor from inhibitory to excitatory. And so this GABA-A receptor is a type of ligand-gated chloride channel, but invertebrates are really cool in that they have a wide range of other types of ligand-gated chloride channels. So mollusks like squid have glutamate, acetylcholine, and dopamine-gated chloride channels. As these are all very functionally similar to the GABA-A receptor, if the GABA hypothesis is true, these other ligand-gated chloride channels will likely be similarly affected by high levels of carbon dioxide. And so in my research, I also wanted to look at whether these other ligand-gated chloride channels present in mollusks may also be involved in behavioral changes at high CO2. So to address these aims, I used a pharmacological approach using 2 different drugs. So the first I used is gabazine. So gabazine is a GABA-A receptor antagonist. It blocks those GABA-A receptors. This is the main drug that's been used in studies in fish to test the GABA hypothesis, and is also the only drug that's been used in those few studies testing the GABA hypothesis in mollusks. But the problems with using gabazine is that its action has been determined within vertebrates like fish. It's not very well known the action of gabazine within invertebrates like mollusks, and it's really unknown. So we think that gabazine does block the GABA-A-like receptors that are in mollusks, but it's very unknown. It hasn't been tested the specificity. So we don't know if it's also going to act on other receptor types, which means the conclusions that we can draw from only using gabazine are quite limited, and why I decided to use a second drug. So I chose picrotoxin because it's structurally unrelated to gabazine, and it's been much better studied within mollusks. So research shows that picrotoxin selectively blocks all the molluscan ligand-gated chloride channels, the GABA-A receptors, as well as those glutamate, acetylcholine, and dopamine-gated chloride channels. So using both of these drugs, I could look into what receptors are involved. So for example, if I find a similar effect of gabazine and picrotoxin, provides more robust support for the GABA hypothesis because the GABA A receptor is that common target of both of these drugs. But if I find an effect of picrotoxin but not gabazine, it suggests those other ligand-gated chloride channels, other than the GABA A receptor, may be involved, as only picrotoxin targets those receptors. So for this squid, after this work, I caught squid from the wild after acclimation. I put them individually in treatment tanks for 7 days and exposed them to either current day or elevated levels of carbon dioxide projected to occur at the end of the century following the worst case scenario. I then did drug treatment across 2 experiments. So the first experiment, I exposed squid to sham, a type of placebo, or gabazine for 30 minutes. And in the second experiment, to sham or picrotoxin. Then directly after drug treatment, I did a behavioral trial. So I placed a squid in a square tank and they had a mirror along the entire length of one wall. And so the squid react to their mirror image as if it's another squid or a conspecific. So here I was looking at visually mediated conspecific directed behaviors. When I analysed the behavioural videos, I defined a zone called zone A, which is just that 3 centimetres closest to the mirror. And so I measured both exploratory and aggressive interactions with the squid with their mirror image, as well as used tracking software to measure activity levels, and then use Bayesian regression models to statistically analyse the results. So overall, across both experiments, I found that the squid had increases in both exploratory and aggressive interactions with their mirror image at high levels of CO2 compared to the current day levels. And the squid exposed to elevated carbon dioxide were also much more active than those exposed to current day levels of CO2. Okay, so for the pharmacological results, I'm just going to show a couple of behaviours to illustrate what I found. So firstly, we're looking at the gabazine experiment, and you can see this graph here are the results. So on the y-axis, we have the treatment groups. So the squid exposed to either current day or elevated CO2, followed by treatment with either sham or gabazine. And the x-axis is the behavior. So in this case, I'm looking at a behavior which was the time the squid spent in zone A, that zone closest to the mirror. And you can see an effect of CO2 with the sham-treated squid exposed to high CO2, spending more time in the zone than the squid exposed to current day CO2, indicating there may be an increase in exploration and attraction of the squid for their mirror image. This caterpillar plot now shows the same results, but just in a different format. So the y-axis, we're seeing the different types of effects, and the x-axis is the effect size as a median fold change. So this vertical dashed line is at 1-fold change, no difference. If you have the point line to the left, we're going to see an increase in the effect, and to the right is a decrease, with the numbers representing the percentage probability. So here we have a 95.9% probability that elevated carbon dioxide treatment is increasing the time spent in zone A. So we have very high confidence in this effect. In the second experiment, we also have confidence that high CO2 treatment is increasing the time spent in zone A. Then if we look at the drug treatment, we can see we found no evidence for gabazine having an effect on this behavior at current day CO2, but in squid exposed to high CO2, gabazine decreased the time in zone A to levels seen in the controls. Now, if we compare the effect of gabazine at current day CO2 to the effect of gabazine at high CO2, you can see we have high confidence that gabazine is having a different effect at the different carbon dioxide treatment conditions. So this different effect of gabazine suggests that altered GABA A receptor functioning underlies this behavioural change, supporting the GABA hypothesis. Now, with picrotoxin treatment, we saw an increase in this behaviour at current day CO2, but no evidence for an effect of picrotoxin at high CO2. And similar to gabazine, we have evidence that picrotoxin is having a different effect across the carbon dioxide treatment conditions. So the different effect of both gabazine and picrotoxin across CO2 provides more robust evidence for the GABA hypothesis in the two-tone pygmy squid. And these results also suggest that other ligand-gated chloride channels may also be involved. So another behavior I looked at was the time until the squid had their first exploratory interaction with their mirror image. And you can see that in both experiments, I have evidence that this time decreased at high CO2. So the squid exposed to elevated carbon dioxide conditions interacted with their mirror image much faster than those exposed to current day CO2, again indicating there may be an increase in exploration and attraction of these squid for their mirror image. So gabazine treatment increased this time at current day CO2 and also increased it at high CO2. And we have no evidence of gabazine having a different effect across the carbon dioxide treatment conditions, suggesting that the GABA-A receptor is not involved in this particular behavioural change. Picrotoxin treatment decreased the time at current day CO2, but increased it at high CO2. And we have very strong evidence that picrotoxin is having a different effect across the carbon dioxide treatment conditions. So this opposite behavioural effect of picrotoxin suggests that those ligand-gated chloride channels, other than the GABA-A receptor because of the results of gabazine, may be involved in this behavioural change at high CO2. And this was a really exciting finding because it's the first pharmacological evidence of these other receptors that function similarly to the GABA-A receptor being involved in these behavioural changes. So overall, from this pharmacological work, I found high CO2 increased conspecific directed behaviours and activity levels of the two-tone pygmy squid. Previous studies have only used gabazine within marine invertebrates. Now, using both gabazine and picrotoxin, I provided more robust support for this GABA hypothesis in a cephalopod mollusk. And using both of these drugs, I could also show that other ligand-gated chloride channels are likely also involved in these behavioural effects, suggesting that the effects of high CO2 on neurotransmission may be more widespread than was initially thought. Okay, so this work has really focused in on the GABA hypothesis and the theory behind the GABA hypothesis. But it's likely that other mechanisms that are yet to be identified are also involved. And it's also true that the nervous system mediates not only behavior, but also physiology. And so the next thing I did is use transcriptomics to use a more holistic approach, looking at all of the genes expressed in the selected tissues to provide a better idea and perhaps develop new hypotheses for the mechanisms underlying both behavioral and physiological responses to ocean acidification. So the overall aim of this transcriptomic work was to investigate the molecular response of the central nervous system and the eyes of the two-tone pygmy squid to high CO2, and use this to identify potential mechanisms within the nervous system underlying these behavioural and physiological responses to ocean acidification. So for this work, I used the same squid from this pharmacological experiment. So the squid exposed to the current day or high CO2, only those sham-treated squid were used. After behavioural trial, I euthanized the squid and dissected out their central nervous system in the eyes and used the RNA from these tissues. So firstly, I used long-read PacBio isosequencing data from 4 samples, so 1 tissue type from each carbon dioxide level, and assembled a reference de novo transcriptome assembly and annotated this assembly as well. I then used the short-read RNA sequencing data from 40 samples, so 10 of each tissue type at each CO2 level, and mapped this against the reference transcriptome assembly to get gene level counts. I then did differential expression analysis, looking at which genes are significantly differentially expressed between current day and high CO2 for both tissue types. And we know that not only changes in expression of single genes, but also small coordinated changes in expression of genes that belong to the same functional category or biological pathway can also alter phenotypic expression. So I did another analysis called gene set enrichment analysis to look at these small coordinated changes in expression. I'm just going to focus on the differential expression now for time, but if you have questions about the methods in this or other results, please have a chat to me afterwards or get in contact, because obviously a lot of work went into this, which I don't have time to go into now. Okay, so overall, I found 25 significantly differentially expressed genes between current day and high CO2 within the central nervous system, and 8 differentially expressed genes in the eyes, with 2 being significantly upregulated in both tissues. So you can see it's a relatively small number of differentially expressed genes, but it is fairly consistent with previous studies looking at differential expression across CO2 in the olfactory system of fish. And so when I looked into the functions of these differentially expressed genes, as well as those functional categories identified from the gene set enrichment analysis results, I identified 3 top functional categories. So one of them is neurotransmission. So I found many genes involved with neurotransmission across multiple different types of neurotransmission, supporting those pharmacological results that not only is GABAergic signaling affected, but also other types of neurotransmission. And I also found differentially expressed genes related to the immune response and oxidative stress, which is when there is that imbalance between the production of reactive oxygen species and the antioxidant defense mechanisms, which can lead to cellular damage. So these findings really support a growing evidence that high CO2 affects the immune system and the oxidative stress of mollusks. And I've shown that this is also occurring in the nervous system. So I just want to talk about some of the differentially expressed genes involved in neurotransmission that are also associated with behavioral changes. So I found upregulation of this gene SYVN1B, which codes for an E3 ubiquitin ligase, which is critical for the degradation of GABA-A receptor subunits. I also found upregulation of this gene here, which codes for a transporter which packages up monoamine neurotransmitters into synaptic vesicles prior to their release as part of neurotransmission. And I saw upregulation of monoamine oxidase A, which degrades those monoamine neurotransmitters. So I'm finding differential expression of 2 key genes involved in monoaminergic neurotransmission. And this is a neurotransmission mediated by dopamine, serotonin, norepinephrine, and epinephrine. And monoamines are known to be involved in multiple different types of behavior. In particular, changes in expression of these 2 genes here are known to change aggression, as well as depressive and anxiety-like behaviors and activity. And that's particularly interesting if you remember my behavioral results, which was from the same squid, that we saw increases in aggression and activity at high CO2. I also saw differential expression of some genes involved in glutamatergic neurotransmission. So one of these was upregulation of FOLH1, which codes for an enzyme which synthesizes that neurotransmitter glutamate. And glutamate is known to play a critical role in learning and memory. And this gene here in particular, FOLH1, changes in its expression have also been linked to learning and memory behaviors. So I've just shown a few genes here, but overall we found differential expression, and this was small coordinated changes of genes belonging to a variety of different types of neurotransmission at high CO2. And so this pharmacological work, as well as this transcriptomic work showing molecular signatures for an effect of high CO2 on multiple different types of neurotransmission, showing that these effects of high CO2 may be more widespread than this initial work, which is really only focused on that GABAergic neurotransmission mediated by the GABA-A receptor. Okay, so from this work, as I said before, I found effects on these 3 main functions. And from this, I developed a mechanistic model hypothesizing how high CO2 could be affecting these functions and how they could interact within the nervous system of the 2-tone pygmy squid to drive both behavioral and physiological changes. So just really quickly, I found evidence of altered neurotransmission, as I talked about. This could alter downstream signaling, altering behavior. But changes in neurotransmission can also affect physiological processes, one of those being the immune response. And I found molecular signatures for changes in the immune response at high CO2. I also found evidence for oxidative stress at high CO2. And it's known that the nervous system is especially vulnerable to oxidative stress. So if oxidative stress is being induced, this could cause damage within the nervous system, altering neurotransmission, then having downstream effects on both behavior and physiology. So from this transcriptomic work, I assembled a reference transcriptome, which will hopefully be really useful for other cephalopod researchers. And I'll be making this publicly available when I publish these results. I found molecular signatures for changes in 3 top functions: neurotransmission, immune response, and oxidative stress at high CO2, and hypothesized this mechanistic model of how they could interact within the nervous system, driving both behavioral and physiological responses. And this work really shows that the effects of high CO2 on the nervous system are likely not occurring in isolation. And so we need to account for these interactions when we look at the mechanisms. It also shows that ocean acidification likely not only affects behavior, but also physiology. And to date, mechanistic work has really focused on those mechanisms in the nervous system underlying behavior. So it would be important to move forward also looking at the nervous system's role in physiological changes. Okay, lastly, so because I have behavioral data as well as gene expression data from the same individual squid, I did a correlational study to do a more direct assessment of the mechanisms or molecular mechanisms underlying these behavioral changes at high CO2. So I used the gene count data from my transcriptomic work. And what I did first is WGCNA, a weighted gene correlation network analysis. And what this does is it constructs a network, and then it detects modules of genes that are closely clustered together based on their gene expression profiles. So to look at this simplistically, we can see a network that has been constructed with each node or each circle being a different gene. And those genes that have a similar gene expression profile are clustered together, creating modules, and each module is labeled by a different color. And in fact, this analysis, WGCNA, does label different modules with different colors so we can recognize them. So then what I did for each of these detected modules is calculated the module eigengene, which is the first principal component of that module. And it represents the gene expression profile of all the genes within that module. So the purpose of doing this is to deal with the multiple comparisons problem of having a large transcriptomic data set. It means that instead of correlating each of about 50,000 different genes with each behavioral trait, I can now correlate each of 20 to 30 module eigengenes to each trait. And so that's what I did next. I correlated these module eigengenes with the carbon dioxide treatment, current day or elevated, and with each of my behavioral traits. So as a reminder for the behavioral results, I found increased activity and increased conspecific exploratory interactions in the squid. So to do this correlation, I used CCA, canonical correlation analysis, which is a multivariate technique. It means that instead of doing a separate two-way correlation between each module eigengene and each trait, I could do it all together in one analysis. And so this analysis allowed me to identify which modules of genes are correlated with which trait, so carbon dioxide treatment and which behavioral trait. Then lastly, what I did with this is I've identified those modules of genes that are of interest, and then I could look into which of those are hub genes. So these are the genes that are most interconnected within the module and the most highly correlated with the traits of interest. So essentially here, I'm finding those genes that are the most correlated with carbon dioxide treatment and with each behavioral trait. Okay, so from this analysis, I found a total of 230 hub genes highly correlated with carbon dioxide treatment within the central nervous system, 25 genes highly correlated with carbon dioxide treatment in the eyes, and 14 hub genes in both tissue types. But I'm not only interested in these genes, but those that are highly correlated with both carbon dioxide treatment and a behavioral trait, because it suggests that those genes are potential molecular drivers of this changed behavior at high CO2. So looking in the central nervous system, I found 158 genes were highly correlated with carbon dioxide treatment and also activity, suggesting these genes might be driving that increased activity we see at high CO2. 6 genes in the central nervous system were highly correlated with CO2 and the number of exploratory interactions, suggesting those genes might be driving that increased exploration at high CO2. In the eyes, there were 6 genes highly correlated with activity and CO2 treatment, and then of those genes highly correlated with CO2 in both tissues, 1 was highly correlated with the number of exploratory interactions in the CNS, 3 number of exploratory interactions in the eyes, and 1 in the eyes for activity. So these genes are the ones that I'm really interested as potential molecular drivers of the behavioral changes at high CO2. And when I looked into the functions of these genes, I found an overwhelming molecular signature for neurogenesis. So neurogenesis is the formation of new neurons. So I'm just going to talk about the central nervous system for time, but looking in at these 158 genes potentially driving increased activity at high CO2, to make sense of them, I did a functional enrichment analysis. And I found a significant enrichment for cell cycle and cell migration, as well as many genes involved in all of the processes involved in neurogenesis. So neurogenesis is a multi-step process. It involves the production of new neurons, but also integrating these neurons into existing neural circuits. And I found genes involved with all of these steps of neurogenesis. Of those genes in the central nervous system that are potentially resulting in the increased exploration at high CO2, I found it included CDK10, cyclin-dependent kinase 10, which codes for a protein kinase, which is really critical for neurogenesis. Interestingly, I also found the same gene within the eyes as a potential regulator of increasing exploration at high CO2. And so overall from this analysis, I'm finding this molecular signature for changes in neurogenesis at high CO2, potentially driving the behavioral changes. So why could this be happening? So a lot of research, mostly in mammals, but also a little bit in marine invertebrates like cephalopods, have shown that changes in the environment can drive changes in neurogenesis. And that can result in changes in neural circuitry, altering the structure and function of the nervous system. And that can go out to have functional effects, for example, altering behavior. So for example, a lot of research has shown that positive experiences, such as exercise, can increase neurogenesis and then go on to increase cognitive performance and decrease anxiety. So it's thought that changing neurogenesis is a really important process, allowing animals to constantly adapt to an environment that is always changing around them. So in the case of the squid, it's possible that high levels of carbon dioxide are inducing changes in neurogenesis, allowing the squid to coordinate responses, including behavioral adjustments, to deal with this change in the environment. But we're yet to find out whether these changes could be adaptive or possibly maladaptive. So overall, because I had this gene expression data and behavioral data from the same individual squid, I could do this more direct assessment of the molecular mechanisms underlying the behavioral changes at high CO2, and identified neurogenesis as a potential key driver of these behavioral changes under ocean acidification. So as a quick recap from this research, I just want to tie it together. So firstly, my pharmacological work identified or found more robust support for the GABA hypothesis in a cephalopod mollusk, and I also found evidence for the involvement of other ligand-gated chloride channels. My transcriptomic work also found molecular signatures for changes in GABAergic signaling, as well as neurotransmission mediated by ligand-gated ion channels, including anion and cation channels, and G-protein coupled receptors, which are some of those results from the gene set enrichment analysis I didn't have time to talk about. And I found molecular signatures for changes in the immune response and oxidative stress at high CO2. This last study, correlating behavior with gene expression, I found neurogenesis as a potential key driver of behavioral change at high CO2. From my transcriptomic work, I didn't have time to talk about this, but I also found molecular signatures for altered neurogenesis at high CO2, supporting that correlational study. And in my correlational study, I also found genes as potential molecular drivers of behavioral change involved in GABAergic signaling, as well as the immune response and oxidative stress, supporting those transcriptomic results. So overall, the research in my thesis has really had an exploration into the mechanisms underlying these biological responses to high CO2 in marine invertebrates, and I used a cephalopod mollusk to do this. And I found that ocean acidification, high levels of CO2, are likely inducing a suite of changes in the nervous system with a complex assortment of mechanisms underlying those responses to ocean acidification. And this complexity in the mechanisms with multiple pathways involved could explain why there is such variability in the responses to high CO2, with potentially some mechanisms being more predominant in some taxa and underlying specific behaviours. So providing this increase in understanding of the mechanisms underlying these responses to ocean acidification will really help us improve predictions of how marine animals will respond as climate change, and particularly ocean acidification, progresses. But we're yet to find out whether the nervous system could adapt as climate change progresses. With that, thank you very much for listening. I'd like to thank everyone who's been involved in this research. So everyone in Townsville at JCU who's helped with the experimental work, as well as helpers with the lab work and all the molecular work. And of course, a massive thank you to the Marine Climate Change Unit here at OIST, led by Tim Ravasi, who have been so welcoming and helped me so much with my research. And in particular to Roger, who has been an amazing mentor and helping me with all the transcriptomic work. I'd also like to say, so these are the 2 publications from my thesis so far. I'm currently finalising the molecular work papers, so keep an eye out for those publications. And I'm also around for 2 weeks around OIST, so I'll be here if anyone wants to get in contact for a chat or meet about anything at all. So yeah, thank you very much.
Thank you very much, Jodi. It was a great talk. I already saw this talk now 3 to 4 times, but I really like it more every time I see it. So congratulations. So any question? So if there is question from the floor, please go ahead. I cannot see you guys, so.
Yeah, we have one question here. Yoppa's bringing over the microphone.
Hello.
Hi.
Thank you so much. The talk was really beautiful, really clear. Yeah, it was awesome. I have like a few questions. So sorry, I'm Charlotte. I'm a new postdoc in the LUSCOM unit.
Oh, nice.
So nice to meet you.
Nice to meet you.
So I was wondering, is it possible that picotoxin has impact on chloride channels in other parts of the body, like muscles or gut, or yeah?
Yeah, cool question. So these ligand-gated chloride channels, I mean, are predominantly in the nervous system, but of course, in cephalopods, the nervous system is so decentralized, it's throughout the whole body. And so we can't differentiate whether this is in the central nervous system or is it diffuse throughout the nervous system. I had some ideas of using a drug that cannot cross the blood-brain barrier, and so that would help answer some of those questions. But yeah, exactly. So we don't quite know. I don't think it would block chloride channels that are not these specific ones in the nervous system, so not those ones that you'd get in muscles and things, but it would be in those, you get those, some of those ligand-gated chloride channels, say in the nerve, like a neuron to a muscle connection, it would be affecting those as well.
Cool, thank you. My other question is kind of towards the end of the talk.
Mm-hmm.
I was kind of surprised to see when you do the correlation analysis, you end up with a lot of genes, but when you do the differential expression analysis, it's a few genes. So something suggests that these changes are quite small that you're detecting in the correlation analysis to not become like statistically significant in your other analysis. So I guess, I don't know, this idea of neurogenesis, you change the acidity for 30 minutes.
So the acidity was 7 days.
7 days. Yeah, so how long does neurogenesis take? And, you know, how long would you predict that you would need to see the impact of this?
Cool. Yeah, good question. So the 7 days, just firstly to talk about the 7 days. So we have found behavioural effects as quick as 5 days. So 7 days is definitely long enough. And with these squid having a really short lifespan, it's actually like about 25% of their adult lifespan. So relative to the short lives of these squid, it's actually quite long. But in terms of neurogenesis, we can definitely see neurogenesis fast enough. So a common method of detecting neurogenesis is this BRDU labeling of cells, and commonly people will inject it and then a few hours later euthanize the animals, section them up and look for BRDU staining. So you can definitely see it within a few hours, definitely a day. So I think 7 days would very likely be enough time for these squid to have neurogenesis happening.
That's really awesome. Thank you.
Yes.
Sure, okay. Thank you for the talk. I was wondering about the GABA hypothesis.
Yes.
So, you know, naively, if we, you know, change the concentration of CO2, we're changing the ionic concentration inside the, you know, the extracellular space in the brain, and that's gonna affect every voltage gated channel in every neuron. So what's so special about GABA?
Yeah, that is a great question. I think the GABA hypothesis is a lot of unknowns. It was mainly based off the idea that these changes, bicarbonate and chloride, link perfectly to those ions already being that flow through the GABA A receptor, but of course it could affect other receptors that have different ion flows. But also the idea that previous research has shown that GABA A receptors can have the switch in function from inhibitory to excitatory, so during development in mammals, for example. So it's known that this function is possible to switch, making it a more likely hypothesis, I guess, is what this initial work was focused on. But we really, obviously it relies on changes in concentration that are different in the extracellular fluid compared to within the neuron. So we have this change across the neuronal membrane, and it's actually not really known is that happening. So there has been one study in fish that has measured changes in bicarbonate ions. I think it was intracellularly, extracellularly, and then they did calculations to show that it's possible that this change happens across the neuronal membrane. Marine invertebrates, nothing's been done, and we also don't really know the permeability ratio of bicarbonate and chloride ions, which makes those calculations quite difficult. Yeah, so it's really just based on this initial idea that it seems likely, it seems possible, but of course, if you're having these changes in ions, there's so many other effects that could be happening. Like one of them is changes in bicarbonate ions can affect potassium channel regulation, and so then that could have lots of effects. And I mean, even with this gene set enrichment analysis, I was finding effects of CO2 on ion channels that are important for action potential generation, for the release of neurotransmitters, some of those processes that is really just required for all neurotransmission to occur and is not specific to types of receptors. So I think it could really be having a much more widespread effect. And whether that's because of ion concentration changes, we still need more research really to make that link.
Yeah, can I add something on that to Sam? I don't know in cephalopod, but we try to measure this in fish, and it's very tough.
Yeah, the chloride ion is like pretty much so low that it's at the limit of detection of the mechanism of how it's like the machinery that detects it. So it's really difficult to get measurements of those chloride ion concentrations as well to see if this is what's happening. Plus the fish and the squid are very small, so that makes it even more difficult. Sorry, Jodi.
Follow up. Is it known what the change in the CO2 in the does to the extracellular ion concentration in the brain?
I mean, not really. So there's research looking at hypercapnia effects, and if we're talking about cephalopods, they've shown increases in bicarbonate ions at high CO2, but higher than ocean acidification scenarios. There's not much at this ocean acidification level. And as far as I'm aware, nothing within the fluid, like the cerebrospinal fluid or within the brain. So yeah, there's a lot of gaps in our understanding of how the theory of how it all ties together. But I guess a lot of it has just kind of jumped to what is actually happening, because obviously climate change is imminent and we're trying to understand the whole animal effects. But I think that would be very, very interesting, getting into more of the specific details of how this is actually working.
There is any other question from the floor? There is no question from the
I have one more. Sorry, Tim.
Yeah,
sorry, Billy has a question.
Is there any understanding of the actual ecological consequences of these behavioral changes? Have people studied this, or is it not really known?
Yeah, cool question. And squid, we don't know. Short answer. Yeah, I don't know of any studies that have really taken these lab tank experiments set up in squid into the wild. There's obviously been some studies, I guess, like with your lab group, with the volcanic seep work, finding changes in behavior in fish. at volcanic seeps, which have naturally acidified water. They're finding changes in behavior at those compared to nearby control level of CO2. And there's also those experiments of, say, raising these fish work in the lab at different CO2 levels, finding behavioral changes, and then say, for example, increased boldness and increased activity, which we would hypothesize you're more likely to get predated on, right? Release those fish into the wild and come back and look at mortality, and they do find that the high CO2 fish are more likely to be dead, to be gone. So, I mean, there's this idea that it has an impact on animals in the wild, but a lot of this marine invertebrate stuff, specifically cephalopod stuff, hasn't been transferred into what's happening in the wild. We can only make guesses of how this tank work will translate. Were there any Zoom questions, Tim?
No, not from the Zoom people. So I have a question, maybe because I forgot, Jodi. So you tested only 2 phenotypes, right? So you tested only the activity and the mirror experiment, correct?
Yes, in this experiment, yeah. So the activity levels and then a range of different behaviors to do with the interactions with the mirror. So there were like 6 or so behaviors
okay,
with that.
6. Okay. That's what I forgot. It's not only one.
Yeah.
Okay.
Yeah, good. So with the exploration was this thing where the squid go up to the mirror image with their arms and they just touch the mirror. And so I was looking at the latency until that happens, the number of times they touch the mirror. And there was one other, and then with the aggressive interactions where you see them go up to the mirror and full on, they change colors, they attack the mirror like at high speed. Similar things, latency till that happening, the number of times they do that. And then also this zone A stuff, the number of times they enter and exit that zone closest to the mirror, as well as how much time they spent in that zone. And then for activity, I did 3 measures. So I did the distance moved, the average speed, as well as the percentage of time spent active or moving. And all 3 of those showed the same results. So
yeah, okay,
as a whole, activity is increased, but I had 3 measures to get to that. Yeah.
All right, great, thank you. So any other question from the floor, from the Zoom? Doesn't seem like it. In the room, no? Well, if not, so I think it was a great talk. Again, Jodi, great job for your PhD. To everyone, Jodi, as she said, she will be with us for the next 2 weeks, sitting around where my unit is sitting. So feel free to stop and talk to her and ask questions and support, okay? And thanks again, Jodi.